Half a day everyone, my name is Sheena Gayos and my capstone presentation is on the role of sensory hairs in hermit crab antennae in response to chemical stimuli. In humans, the act of olfaction or smelling is simply drawing in air into your nasal cavity. However, hermit crabs are a little more unique in that they use their external sensory organ, the antennae, in olfaction. Chemoreception is an essential part to any animal as it helps them find food, avoid predators, and reproduce. In this experiment, I will observe the function of sensory receptors on the hairs of hermit crabs and how they sense their environment. With this, I will explore the physiology of the receptors receiving the stimuli and how they send this information to their brain. And with the data I obtain, I will compare the responses between the various scents and chemicals, as well as the action potentials they elicit. With this, I will propose possible lab techniques in investigating the physiology of other arthropods and invertebrates. This type of experiment is important in that not much is known about the olfactory system in terrestrial hermit crabs. More is known about aquatic hermit crabs in that they sniff the water or odorants in the ocean by creating currents between the antennae to trap the fluid in spaces between the hairs during the slow downward stroke, and then they hold this water sample and detect the odors during the slower return stroke. However, in terrestrial species, they have a shorter antenna and they use air flowing outside the antennae to observe any odorants. The Cenobita cavipes, or the land hermit crab, is known as the tuk-tuk here in Guam, and it is a primarily nocturnal animal and supertidal as it prefers access to both land and water. Its body consists of a curled tail, and it enables them to fit inside a spiral shell, which without it, they are extremely vulnerable to predators and drying out. They are a scavenging animal in that they primarily feed on whatever washes ashore, such as algae, fish, or rotting wood, and they use olfaction in finding this food. And one of the first things you notice about hermit crabs is that they have two pairs of antennae that are constantly flicking. The long pair is used for touching and feeling, and the short pair is for smelling and tasting. On each of the antennae are many sensory hairs, and they're also known as setae hair sensilla or ascletas, which are also located on the exoskeleton. On each of the hairs, there are simple sensory receptors used for olfaction. So this constant flicking of antennae is their way of sniffing the air around them. Here is a microscopic view of the antennae. You can see that there are many sensory receptors and hairs on the antennae, which all help in olfaction. Some physiology about the sensory receptors. In order for the hermit crab to detect the odors, the odorant must first be dissolved in some sort of medium. And each single sensory hair contains cilia with hundreds of bipolar neurons, and the axons of these olfactory sensory neurons project into the brain's primary olfactory lobe. As we've learned before, the bipolar neurons serve as an intermediate between the stimuli and the nervous system. This allows the hermit crab to show a narrowed response to a single stimuli known as downregulation, or they can elicit a broad response to multiple stimuli, such as how they block out the scent of the ocean in order to better detect the scent of a potential predator. Here is the neural anatomy of the hermit crab. You can see the olfactory lobe takes up a significant portion of the hermit crab brain. Here is another fluorescent X-ray of the hermit crab brain. The olfactory sensory neurons from each of the ascletasks converge into one antennal nerve leading into the olfactory lobe, and the brain is dominated by both the primary and secondary olfactory centers. They have olfactory glomeruli, which are fundamental processing centers in the olfactory system. And as mentioned before, olfaction is a major sensory modality in hermit crabs. Here is a video I have of the hermit crab, you can notice the flicking of the antennae and how they smell and observe the scents around them in their environment. My question is, how do hermit crabs respond to different odors? This could be from seaweed or a foreign vegetation such as bamboo, ocean water versus a harsh chemical such as chlorine, sand versus a potential predator such as bird urine, and fish versus a foreign food source such as beef. The second question I have is, can these sensory action potentials be generated from different odorant stimuli? Meaning, can we find and observe these action potentials using correct equipment? My hypothesis is that a high response should be seen in the seaweed, chlorine, and bird urine, while a low response should be seen in bamboo, ocean water, and sand. And I do believe that action potentials can be recorded using certain techniques, and the technique I use in this experiment will be electroantenography. It was first discovered by the German biologist Dietrich Schneider in observing how insects react to different pheromones. It primarily works by measuring the average output of insect antennae to the brain in response to odors. So the action potentials are measured in the extracellular hemolymph or invertebrate blood. And with this, the electrodes will be attached to different areas of the antennae. And the measured voltage fluctuations we will observe in the graph is caused by olfactory neurons depolarizing due to the different stimuli. So here is an example of how electroantenography is done. It's usually done on an isolated antennae, but for my experiment purposes, I wanted to leave the antennae attached to the hermit crab to prolong the life of the antennae and the action potentials. Here is the lab method I intend to carry out. Here are my materials and equipment. You can pause the video to read through the whole material list that I have here. And the method I choose to do is, first you should obtain the hermit crab with a functional antennae, which should be partially sedated to prevent any movement of the body and musculature so that we can get a correct reading. Then we should set up the electroantenography equipment and attach the glass micropipettes to the electrodes to the antennae of the hermit crab. It should be on the distal end of the antennae as well as the proximal end of the antennae or its head. Then the hermit crab will be exposed to the different odorants using filter paper that was soaked in each odorant. And with that, we will record the action potential signals, and there should be an interval of at least 2 minutes between each odorant to prevent any cross-contamination. And with that information, we will compare the results with the different odorants. Here is the experimental setup I devised. The 2 electrodes will be connected to the respective antennae, which will lead into an amplifier and then to a computer system with a chart reader or software that can read the action potential signals. Ideally, this experiment will be done within a Faraday's cage to reduce any noise or other electrical resistance from other sources. And the electroantenography signal is, as I mentioned before, a voltage deflection in the graph. And the source of voltage and antennal resistance will be from the olfactory cells on the antennae. The results I expect to see are that there should be a high response to seaweed, chlorine, bird urine, and the fish scent, as well as a low response to ocean water. So there should be a high response shown by this graph here, where you can see a large voltage deflection, and ocean water should elicit less of a response here. As you can see, it is close to the baseline. With this, we can measure how hermit crabs select different odors and if they favor odorants compared to other ones. This information is important because It can facilitate further research and techniques on the neurophysiology of hermit crabs and other crustaceans, and it will also provide much needed information on the olfactory neurophysiology of the terrestrial hermit crab, because as I mentioned before, not much is known about this certain species. And this will also bring light to how environmental factors affect the olfaction in hermit crabs, such as in odor pollution in today's environment. And here are the resources I gathered my information from, and thank you for watching my presentation.